Cell podcast, Glial Cell Fate with Dr. Paul Tesar. Hey, everybody. This is Dr. Daylon James here with Dr. Arun Sharma. Welcome back to the Stem Cell podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, rate us, leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have an old friend, Dr. Paul Tesar, who's from Case Western Reserve University. And he's on the podcast to talk about his research on glial cells and the central nervous system. Of course, we've also got our usual roundup of recent highlights in stem cell news. That's coming right up.
But first, neuroscientists looking for more predictive power in their disease models are increasingly adopting human pluripotent stem cells in their research. STEMCELL Technologies offers products, protocols, and training to support HPSC-derived neural models. Explore their collection of technical videos and webinars on neurological disease modeling by visiting www.stemcell.com/neural-disease-model. I'm actually going to start things off with a bit of ESC and iPSC and organoid news in particular. This is something that I feel like those of us in the US should monitor. If you haven't heard, the NIH just announced that they have created a dedicated organoid development center to reduce reliance on animal modeling. And it's called the Standardized Organoid Modeling Center, which they say is going to be a national resource dedicated to cutting-edge technologies to develop standardized organoid-based new approach methodologies, or NAMs, that deliver robust, reproducible, and patient-centric research findings. So the emphasis there is on NAMs. They specifically said it in their press release, which you can check out. And these new approach methodologies, NAMs, include, of course, organoids, but also organ-on-chips. And this has been a big emphasis as of late for the NIH. This is leaked out in some capacity in the past, and you may have heard before. And even my boss, Clive Svensson, had a little editorial in Cell Stem Cell not too long ago talking about these NAMs and how they are going to supplement animal models, but perhaps shouldn't replace them entirely. And this is, again, a new center at the NIH, a new center that's focusing on a specific type of NAM, that we know and love here are organoids, of course. So the goal is going to be to leverage these amazing organoid technologies that we talk about all the time on the show to enable optimization of organoid protocols. I think this is a good thing since, organoids, because they're not 2D, because they're more heterogeneous than the 2D cultures, tend to be a little bit less standardized. So hopefully an initiative like this can serve to further standardize the production of these organoids, whether it's kidney, like I'm about to talk about, or cardiac, or neural, or whatnot, ultimately for the purpose of, downstream modeling of disease and also for drug discovery, right? One other thing to mention here is that they're allocating $87 million in contracts for the first 3 years. I'm not exactly sure what that means, whether it's contracts that'll be dished out to external investigators to focus on organoid biology. But regardless, I think it's a really interesting, piece of news for those of us here in the States, and especially those of us who are working on organoids. And I think it's just a reflection of how prevalent these technologies and NAMs have become, and that they're being noticed by, of course, the federal level and the NIH. And they're going to continue on growing and getting better. I don't know, Dalen, do you have any thoughts on this?
Yeah, I appreciate the way you started it there in terms of, I think the consensus is that while these NAMs are going to be amazingly useful and complementary, the idea of ditching the animal model altogether is ludicrous. And I know no one's really I don't think anyone has that as the plan. But I appreciate here that we're building out something in terms of this SOM Center. so that, yeah, we can have alternatives. I don't think we're ever really going to be able to get away from the physiology of an animal model, but I like the idea here that they're building something out that can, relieve the need or substitute in some manner. So I appreciate this endeavor, and I hope that they will put out a FOA to try and recruit some, external investigators in addition to the on-campus complement there to help contribute to this resource, because organoids, everyone's got a different recipe, and I really do think that some kind of standardization is going to be essential moving forward.
Yep, absolutely. And on the topic of organoids, that brings us really nicely into our first paper for the Roundup, which is an organoid paper. And it's actually building on the work that we already discussed with Dr. Zhongwei Li. He actually alluded to this paper in his conversation with us a few months ago. The title of the paper here is Spatially Patterned Kidney Assembloids Recapitulate Progenitor Self Assembly and Enable high fidelity in vivo disease modeling. This is a cell stem cell paper. And again, Dr. Lee is from just down the road from me here in Cedars-Sinai, and he's down the road at USC, University of Southern California, here in LA. So I'm not going to go super deep into this, in part because I think it was already discussed ad nauseum by the senior author himself, Dr. Lee, just a few episodes ago. So check that out if you haven't listened to it. But basically, yeah, this is what he was alluding to, the creation of these human kidney progenitor assembloids, or HKPAs, which are developed from pluripotent stem cell derived iNPCs and UPCs, subtypes of the kidney here. And why do they do this? Because current kidney organoids, which have improved dramatically over the last few years, they still don't perfectly recapitulate the kidney's really complex spatial patterning, function. If you look at the kidney, you got those like nephrons and all these really complex tubules and everything else like that. And so if the organoids are not complex enough, that means their applications will be limited as well. The human kidney, it says right here, comprises more than 1 million different nephrons derived from nephron progenitor cells, those NPCs, that can connect to an arborized ureteric progenitor cell, UPC, derived collecting system. So that's what you have to recapitulate in the assembloid model, and current organoid models of the kidney haven't gotten there yet. So here's what they did. They created not just mouse, but also human kidney progenitor assembloids, where these nephrons undergo really great extensive development and actually fuse to this centrally located collecting system. So you actually get that functionality, which is really wild. You should check out some of the images in this paper. They're really quite beautiful and striking. So it's able to recapitulate that kidney progenitor self-assembly process that importantly happens in vivo. And we were just talking about on the news at the top of the roundup here, how we want these organoid models to supplement the in vivo systems, but ideally be advanced enough to recapitulate in vivo biology as well as we possibly can. And that's what these KPAs are able to do. They show dramatically improved cellular complexity and maturity and exhibit a bunch of different aspects of major kidney functions, both in vitro and in vivo. So then they brought it to the disease model, which I think part of the reason why it's in cell stem cell. They modeled human autosomal dominant polycystic kidney disease, or ADPKD, with genome edited in vivo grown human KPAs, and it was able to recapitulate that cystic phenotype that you would expect in that disease, the different molecular cellular hallmarks of the disease, and the crosstalk between the other cell types that are integral to the disease manifestation. So it's a awesome platform. I think it's reflecting not just the power of organoids, but the power of these next level assembloids that are all the rage these days, and enabling all the things you would expect with organoids: high fidelity disease modeling, drug toxicity and drug screening studies, and a foundation for kidney regenerative medicine. So like I said, check out the interview that we had a couple episodes or a couple months ago with Dr. Lee to learn more about this, but really check out this paper. It's beautiful work.
Yeah, I was really looking forward to this during that episode, just seeing the pictures, right, and the videos based on the description of Zhangwei. And they did not disappoint. I mean, bonkers, so beautiful. Particularly like the ex vivo culture of the mouse kidney. I mean, which isn't a crazy technological feat, but it really, I think, illustrates that branching morphogenesis that is a critical piece of nephrogenesis. And that to recapitulate that then with the human cells, I thought was a real feat here. Although I think there is still some way to go. In fact, seeing that the ex vivo culture of the actual kidney, that really drove home how complex these morphogenic changes are, and how dynamic, and the coordination of all the cells that needs to go into that. And as acknowledged at the back end of this paper on the limitations, the human correlates that weren't exactly able to recapitulate that same degree of branching morphogenesis and nephrogenesis, and that's not unexpected, I think, coming from a ES derived origin as opposed to in vivo cells. And I think that really is a critical gap that needs to be bridged moving forward. Not just because of in vivo versus in vitro origin, but the timescales involved between the mouse and human development, I think, are going to be key steps forward and obstacles to overcome to get a real beautifully patterned and not necessarily functional, because you can do a lot with the modeling without actually getting something that's going to work. But as we discussed in that episode, which I think you guys should ring up and listen to, um, part of the idea here is to create a functional surrogate, right? And in order to get there, we're going to really need to get the patterning exactly right, or close to exactly right. So beautiful illustration of the power of the system here, and a clear pathway forward, and how to improve it even more so.
Yeah, I think the scalability is, of course, going to be the next thing here. And that's something that this new NIH center is certainly interested in: the scalability, the reproducibility, the standardization of the models, and the protocols used to actually make these complex assembloids. And we've talked about papers in the past that have done a great job when it comes to the scalability of these really complex organoid types. Like, I always refer back to that Oscar Obulis paper with the vascularized cardiac assembloids, which not just were really advanced, they were advanced and highly scalable. So amenable to whatever downstream high throughput biology you want to do. I'm the high throughput guy. I gotta give a plug to anything high throughput. So, but that's part of the reason I love these advanced organoid models.
Yes, and as you just described there, the complexity of these things can sometimes be, I don't want to call it an obstacle, but I'd say, or even a confounder, a variable that can make the field as a whole challenging, which is what that NIH Center hopes to address, is to move these things less from like art, where there is such a operator specific element, to some real standardized approaches. And I think that this paper is a step in that direction, a very systematic, formulaic approach, and we're going to see those methods be rewritten, I'm sure, in a protocols paper to come. But moving away from this point of the spear, standardized organoid assembloid models for clinical application and disease modeling, etc., I'm taking it back in this story to the basics. 10 years ago, well, we were in early days of this podcast, it was still all the rage just to talk about what are the influences that modulate pluripotency and early stage differentiation. And this is a callback to those days, except with the newfangled tech of today. It's a nature cell biology story from Yarui Diao from Duke University Medical Center. Go Duke, Arun. And this is about transposable elements, right? All right. So these are the parasitic mobile DNA elements that make up over half of the human genome. Pretty scary, and they've really shaped the human genome over eons of evolution. Not just human, all animal. Transposable elements are ubiquitous, and dysregulation of transposable elements can lead to aberrant development, disease, cancer, And for this reason, we, as the host organisms of these transposable elements, have evolved all kinds of cellular or cell intrinsic mechanisms to suppress that transposition activity, them hopping around so much. And that involves the epigenetic modifications of the transposable element sequences that lead to heterochromatin formation and silencing. Um, but in addition to that kind of negative pathologic influence, transposable elements can also act as cis-regulatory elements, so-called enhancers, that can modulate cell type specific expression of genes. Um, but the missing link here is, unlike gene expression more commonly, which we've talked about in the last few years a lot, about the input of mechanosensing and mechanical inputs on gene expression, but the idea of how transposable elements respond to any environmental stimuli, much less mechanotransduction or mechanical stimuli, is relatively uh, undervalued or understudied. Um, but as I said, the idea that they do respond is implicit, right? Because they make up over half of the human genome, right? And mechanical forces, as we've discussed ad nauseum on the show, play a critical role in development and cell differentiation. And they do this oftentimes by activating this hippo YAP mediated mechanotransducers, right? So in this story, uh, Diao's group here shows that there's this input of matrix and the mechanical properties of cells that actually regulate hundreds of transposable element subfamilies in human cells. And then they also identify a subset of these transposable elements that are mechanoresponse enhancer elements. Okay, so specifically responsive to mechanical stimuli in an enhancer fashion. And mechanistically, they show, as not unexpectedly, that these mechanoresponse enhancer elements are governed by YAP, and going deeper into the mechanism, show that this YAP recruits BRD4 and CTCF um, to modulate the interaction between these transposable element mechanoresponsive enhancer elements and human genes. And here's where the tie-in with stem cells and differentiation comes in. They show that there's this specific subfamily, this LTR7 element, that actually acts as a distal enhancer for this gene, FAM189A2, obscure. But by modulating the function of that gene, inhibits definitive endoderm differentiation. So, like, a crazy amount of mechanism, and connect the dots in this story to link mechanotransduction to stem cell differentiation, or the inhibition thereof, via these transposable elements. So yeah, of course, like, this is a deep basic story, and the firsthand may not jump to mind how we're going to apply this in our everyday work. But I think for me, the real key was conceptually just to incorporate this other input, right? This other wrinkle, which is ubiquitous in our genome, acting as an enhancer. And of course, as all these studies typically do, uh, illustrating the function here in the context of stem cell differentiation, of course, raises the question of how these transposable element MREEs may act across the board in our own systems of interest. For you, the heart; for me, reproductive axis. So a lot to think about there. How do we begin to flesh out that question in our own systems remains to be seen. But I think definitely food for thought. Arun, what do you think?
Yeah, this is a extremely deep mechanistic dive by our friends at Duke University. Go Blue Devils. This is a deep dive on something that we kind of already know and have known for years, and that mechanotransduction is critically important for stem cell differentiation. I mean, that's big picture. That's about the extent to how far I've dove into this area of study. But they're really talking about how these transposable elements are doing the deed here, and I like their focus in mesoderm differentiation in particular. And we're talking about how there could be transposable element independent and dependent processes to regulate mesoderm differentiation. So I think there's a lot of work that still has to be done to flesh out the details of this. I mean, this is a segment of the genome that's it's not super well understood in terms of the role of these transposable elements in regulating distal enhancers and all this kind of stuff. There's actually a big focus of what my postdoc mentors were working on, the John and Cricket Seidman, the role of distal elements in regulating mesoderm differentiation. So I'm sure they've taken a look at this paper. But yeah, big picture, wrapping it up, just kind of the big picture for those of us not in this field is just a reflection of how important mechanic transduction is in stem cell differentiation. And a familiar face and a familiar protein in YAP is, of course, regulating that. YAP is also really well known in its role in cell proliferation, but in mechanic transduction in particular. So it's cool to kind of take a deeper dive into this.
Yap, like went the center of it all. But yeah, this, I mean, it kind of got me into, I don't call it existential crisis, but I went just thinking very deeply about this, just because, yeah, I mean, we know mechanotransduction is important for many facets of differentiation, and transposable elements are literally everywhere in the genome. So just like on a kind of like evolutionarily, existential, teleological, I don't know what kind of level, it's crazy to think how many potential influences there are in our biology that can affect, our genetic heritage. And then maybe that is a little bit too deep, but it just was overwhelming for me from an existential perspective on just the randomness of it all, and how many things can affect our evolutionary purview. So a story that went from deep mechanism for me to philosophy, Arun. So I came out of here with a bit of a headache, but definitely thinking.
Yeah, it's we love talking about these Evo Devo evolutionary development angle stories. And if you think about it, we have 99% genetic similarity to our closest relatives in the animal kingdom, like chimpanzees, and for example, and it's that 1% that could be dictated by these transposable elements and other distal regulatory factors. So a lot to think about for sure. Sticking in the realm of mesoderm differentiation or mesoderm in general, I'm talking about another cell stem cell paper here that's focused on the heart, my favorite organ. The title of this paper is Targeting Lysozyme 2 in Endocardium Promotes Rapid Recovery by Modulating Remote Injury Signals. And this is a stem cell adjacent story, but is a reflection on one of my favorite topics, which is cardiac regeneration. And I talked about it before on the show. And another Duke connection here. Cardiac regeneration is kind of how I got into the stem cell and the heart research field. When I was at Duke as an undergrad, I had the opportunity to connect with Dr. Ken Poss, who's, of course, an expert on all things heart regeneration in the context of the zebrafish. Zebrafish, of course, famously can regenerate their hearts, but adult mammalian hearts can't regenerate, and that just kind of got young Arun thinking and got me excited about the field in general. So enough reminiscing. We'll focus on the current paper, but that's what it's all about, really. Adult mammalian hearts are not regenerative, right? And so a lot of studies that are examining repair and post-regeneration, post-MI, post-myocardial infarction, have focused on cardiomyocyte proliferation, right? The proliferation of the remaining heart muscle cells after an adverse event, such as a heart attack. And also, these studies have looked at fibroblasts. I mean, some folks like Deepak Srivastava and other folks are interested in transforming some of those fibroblasts in the heart into functional cardiomyocytes after myocardial infarction to help restore the function of the heart after a heart attack. I mean, there's the results have been mixed, but that is underway, and those are approaches that are being investigated. Here, they're taking a different mechanistic angle. They're looking at a really high expression of a protein called lysozyme 2 or lys Z2 in injured mouse hearts at both the local injury sites and at distal remote zones. They found that the expression of LYZ2, lysozyme 2, is in endocardial cells, specifically of non-regenerative hearts. So the endocardium is the innermost lining of the heart and is associated with the endothelium and the endocardium, the blood vessels and all that kind of stuff, right? Um, and traditionally, it was known as a myeloid marker, this LYZ2. They actually showed that it functions as an injury-specific positive regulator of lysosomal degradation capacity. That's kind of like the trash can of the cell, right? That actually mediates the pathogenic degradation of the extracellular matrix. So in the case of the heart attack, myocardial infarction, this LYZ2 is functioning as that molecular trash can to clear out some of that nasty leftover matrix, perhaps. And so there's a mechanism here where they found that there's actually an anti-apoptotic benefit to cardiomyocytes when you actually disrupt this LYZ2 lysozyme function in mice. So if you alter it, if you inhibit the function of this protein through knockdown or knockout, you actually get improved survival of cardiomyocytes after the MI and more rapid recovery of these mice. Okay, so anti-apoptotic benefit. And they also had a human endomyocardium experimental model. It's more of an in vitro system that they looked at there just to kind of dive into the mechanism there. So when they knocked out the LYZ2, like I said, or they did a pharmacological inhibition of that lysosomal degradation, it's able to confer enhanced recovery in the injured non-regenerative adult mouse hearts. So it's an interesting mechanism here, interesting observation, where if you target a remote injury response, maybe in a non-cardiomyocyte cell type, like in the endocardium of the situation, maybe you can regulate the regeneration of the myocytes. And that's kind of what they were going at here, the cell type specific interaction crosstalk, right? And ultimately, it's a question that still needs to be addressed. How do we get adult human hearts to regenerate after a heart attack? And maybe there's an interesting mechanism that's focusing on the lysosomes here that we can flesh out a little bit further. I still think, and we've talked about this before, I think the end game is not so much on the regeneration of the existing remaining cardiomyocytes, it's the replacement of the entire heart. Maybe that's a bit of a stretch in a situation of like myocardial infarction where there's some reduction in function, but not a complete loss in function of the heart, like a heart failure system. So maybe it's taking it a bit to an extreme to just swap out the entire heart after somebody has a heart attack. But regardless, I think this is a pretty cool mechanism that they found here.
Yeah, I mean, hey, why not? Swap it out. Swap it out. We got them. They got them coming from the pigs, but the maybe a bit of a stretch. And I think, well, for me, I do agree with you in terms of cell therapy in the heart, which was an early target and biggest target, remains so to this day. Um, that it's a heavy lift, we've learned, right? To deliver cells and to have them be functional, and to how do you mitigate the existing scar, all those things. So there's so many challenges there to the cell therapy that I'm with you that, like, maybe ultimately, pragmatically speaking, it's going to be a matter of doing these xenografts, right? But I think that the reason why that seems like such a ready solution is because approaches like this have not uh, come to pass or have not shown efficacy up to this point. But I'm really excited about this as an idea. It's not brand new conceptually, the idea of drugging the post MI response and mitigating scar and perhaps preserving function. But here, I think they've taken it to a level of proof, demonstratively so in the mouse model at least, with various forms of cardiac injury, exploring the mechanism, proposing kind of a link there. Uh, Although I think there's still a lot of work to do in functionally defining the role of endocardial endothelium there, like how they're mediating the effect. But I love this as an idea, right? You cutting right to open heart surgery and swapping out the heart, but if you could drug it, at least as a bridge, or in less extreme cases, I feel like that's a paradigm that's really well established in our healthcare system. So I love this as an idea, and I would love to see in relatively short order them take this to a large animal model where the scale will be more appropriate and relevant to heart attack that we care about in humans.
Yeah, this is why I'm not a cardiologist or a cardiothoracic surgeon, and my wife, who is a cardiologist, would probably laugh in my face to if I was to recommend just swapping out the heart in the situation of any myocardial infarction. But I'm just a cardiac biologist who likes to dream big, maybe, and I'm just really excited by the developments in xenotransplantation. But yeah, still got to take that mechanistic deep dive, and I think work like this and the work of Ken Poss is still extremely important because just from an Evo Devo perspective, like we were talking about not too long ago, I just think it's so fascinating about, why the mammalian heart doesn't have this capability, but the hearts of other lower vertebrates, quote unquote, have amazing regenerative capacities. So even 15 years later, it's still one of my favorite areas of science to think about.
Well, I'll tell you, Arun, my wife would be very interested in swapping out my heart on some days. That's for sure. Um,
And your brain too, maybe.
for sure, and definitely interested in swapping out my creeping fat, which is my segue to the next story, which is a cell story with creeping fat. First two words of the title, they really suck you in. A little bit of clickbait there. But this is really about Crohn's disease, this article, which is a subtype of inflammatory bowel disease in which chronic inflammation triggers intestinal fibrosis, and thick scar tissue replaces a normal bowel parenchyma, which narrows the lumen there to cause a stricture, right? And this is particularly malignant, maybe not the right word, but it's bad in pediatric patients, and maybe different from the course it takes in adult patients, but up to 50%, between 30 and 50% of pediatric patients develop this intestinal stricture within 5 years of the initial Crohn's diagnosis. And approximately a quarter of them are going to require intra-abdominal surgery within a decade of diagnosis. And a quarter to half of these strictures recur even after these surgeries. And there's no antifibrotic therapies that have been approved to address this fundamental etiology of the fibrosis and causing the stricture. Right. And here's where creeping fat comes in. I never heard of creeping fat before, but it's actually this diagnostic indicator. And what it is, it's this fat, it's an expansion and wrapping of mesenteric adipose tissue around the diseased bowel, specifically in the context of Crohn's. And it's long been utilized as a landmark for surgeons to identify strictures when they're doing the resection of the intestine there, to mitigate. And the creeping fat is associated with recurrence of the stricture, and if you remove or do your best to remove as much as you can of the creeping fat, it reduces the stricture rate. So there's a clear link between this creeping fat and the stricture and severity. Uh, Single cell RNA-seq has been done on creeping fat and shown that it has these networks of pro-fibrotic immune and stromal cells that stimulate ECM deposition, aberrant adipocyte differentiation, and hypertrophy of smooth muscle cells, right? So all things that are going to contribute to this fibrosis and stricture. But the direct contribution of these creeping fat derived stromal cells to the stricture in the context of Crohn's disease and the progression of the stricture not well understood, right? But here I'm coming back to mechanotransduction. Mechanic transduction, mechanical stimulus has long been thought to underlie a lot of disease, right? In terms of the extracellular matrix composition, stiffness in cells can promote fibrosis and progression of fibrosis. And it's been shown also in Crohn's disease that the level of stiffness can exacerbate the condition. And another thing here that's specific to Crohn's is that these strictures, for the most part, they manifest at this ileocecal junction where it's really small, well, relatively small diameter of this valve, this ileocecal valve. And so when the intestinal contents are bearing down on that, it can exert a lot of mechanical stress. Again, the idea there being contributing in the context of Crohn's with this reduced diameter to the fibrosis at this site. So someone who's been interested in fibrosis and wound healing, done a ton of that, Michael Longacre at Stanford, along with Jung Hyun, sorry, their groups mobilized to understand the mechanotransductive element and input in the context of Crohn's. And the hypothesis that they were working on there was that these creeping fat-derived mechanosensitive fibroblasts were promoting the fibrosis in the intestine. And of course, they were promoting it mechanistically by way of this YAP-TAZ-dependent signaling that we just talked about, and we talk about a lot. So the first thing they did is they went into the publicly available database. And I love how so many stories nowadays start out with this. You go into the libraries that are out there that other people who generated, and using these publicly available databases, they generated a Crohn's disease fibroblast atlas in this meta-analysis of scRNA-seq studies. And then they integrated this spatial transcriptomic element and extracellular matrix ultrastructure analysis to show that these mechanosensitive fibroblasts, that they were in these fibrotic niches that are at the interface between the creeping fat and the bowel wall. And then they developed this mouse model. So they went deep on this. This is a cell story not for no reason. They developed this mouse model that reliably generated this creeping fat and had the sequelae of intestinal fibrosis. And using this model, they very deliberately and systematically showed that these creeping fat-derived mechanosensitive fibroblasts, they migrate into the site of the stricture and that they promote fibrosis. And here's the kicker. If they can block YAP-TAZ signaling, they can significantly reduce the fibrosis. So this really runs the gamut, I think, of discovery, taking data that's out there, developing your own tools and models, and then ultimately leading to a druggable target and demonstrating some efficacy of mitigating this fibrosis. So I love this story, similar to the story you just talked about. I mean, we talk about stem cells all the time, regenerative therapies, but drugs work now, and so much more amenable to the way we do things in our healthcare system. YAP-TAZ inhibition, yeah, maybe a bit of a sledgehammer. I'm not saying that this is the approach, but defining the mechanism, tracking the cells, all the details of this story, I think really elevated it and painted a beautiful and complete picture. Arun, what's your take?
Yeah, amazing stuff coming from Dr. Longacre, who some of you may know was on my thesis committee when I was at Stanford. He is the rare surgeon scientist who also is a top-notch basketball player. If you know, and we bring up this story every once in a while, how he was on the 1979 Michigan State national championship team, I believe, as a walk-on. Good friends with Magic Johnson, if you can believe it. But anyways, yeah, great stuff coming from Dr. Longacre. It's not exactly the strong focus of his lab. I mean, they focus more on wound healing, right? But a lot of conserved mechanisms here when it comes to the role of mechanotransduction, like we talked about in fibrosis and that sort of thing. So I think that's kind of why they decided to get out of their wheelhouse. And it was certainly a very collaborative effort here. Really cool stuff, mechanistic deep dive. I think there are a few limitations, and they acknowledge them, that where they were using these single cell RNA-seq and Visium and Xenium studies, they actually had pretty low patient numbers in the studies. So that, of course, it's a limiting factor. And also, the single cell RNA-seq was largely done on biopsies, which don't sample those submucosal layers. So where the fibrosis actually predominates. So a few limitations there when it comes to the technical side of things, but knowing Dr. Longacre, I'm sure they're already addressing these things.
Yeah, and I would argue that this is exactly his wheelhouse. Although clearly, bringing Jong Hyun into the picture was the Crohn's expertise there. But it's all about like the maladaptive healing process and reverting to the way the way Keon heals, bro. The way that we used to be able to do it. I'm just like I picture my insides right now is just like a maladaptively healing wound writ large. And if we could mitigate all that scar formation on my insides, mostly in my heart, then hey, I might live forever. So there's a little bit of magic for you. Johnson can keep his. I'll take that Longacre magic. And that ends the roundup on a high note for me, hoping to live not forever, but to live well as long as I can with minimal scar. And perhaps we'll talk about the scars of life and science just a second. But before we get there, I have a quick message from STEMCELL Technologies, who's inviting you to find a pathway into the peripheral nervous system with STEM-Diff Neural Crest Differentiation Kit. Whether you need Schwann cells, sensory neurons, or sympathetic neurons, STEM-Diff Neural Crest Differentiation Kit enables high purity generation of neural crest precursors from human pluripotent stem cells. Learn more at www.stemcell.com/NCKIT. All right, you guys, on the show today, we have a special guest, an old friend from before I started hosting the show, and a real living legend, I would say, from the founding days of stem cell science, Dr. Paul Tesar, who is the Dr. Donald and Ruth Weber Goodman Professor of Innovative Therapeutics, also director of the Institute for Glial Sciences at Case Western Reserve University, also an all-around great dude and scientist. The Tesar Laboratory sits at the interface between stem cell biology and developmental neuroscience. They use pluripotent stem cells to model neurological development and disease with a specific focus on oligodendrocytes and astrocytes. The interests in the lab are focused on the molecular mechanisms by which cells acquire glial fate and function during development and maintain their functionality throughout life. Paul, thanks so much for joining us on the show today.
Dale and Arun, thanks for having me. I really appreciate both of you. Excited for the conversation today.
Yeah, I'm very excited. I can't believe I haven't had you on more recently, but yeah, really excited to get going here. And I've followed your lab's work, obviously, since we met almost 20 years ago, which is kind of crazy to think of. When we were both funded by the NICEF. In that time, I've been awed by both the output and caliber of your science, but more than that, the creativity that's reflected in your group's approach over the years as like the technology and the capabilities expand. So why don't we start with you giving our listeners a brief overview of what your group is focused on these days?
Yeah, absolutely. So the team is broadly focused on neurodevelopment and neurological disease, but we really approach these topics through the lens of glial cells. And so in particular, as you mentioned, we focused most of our attention on one specialized glial cell type, which is the oligodendrocyte. This is the myelinating cell in the central nervous system. And so we use pluripotent stem cell and reprogramming technologies to understand key aspects of oligodendrocyte development, as well as how oligodendrocyte loss or dysfunction may contribute to neurological disease. And we combine these scalable cellular approaches with high throughput chemical and genetic screening to identify therapeutic targets that might be able to improve oligodendrocyte function or regenerate oligodendrocytes that have been lost. Right, we have a strong translational focus in the lab, and we've been very purposeful about trying to advance our science out into the commercial and clinical sectors in order to reach patients. We have a number of glia-targeted therapies that have advanced out of the lab, and I'm excited to see to see the field move into what may be a new era of glial medicines.
Yeah, that's a great intro because we're going to be talking about a little bit of everything that you just mentioned there. So, starting off, let's actually talk about a very recent study coming from your lab, this cell paper that we actually covered on the podcast not too long ago, titled Transient Gene Melding Governs the Timing of Oligodendrocyte Maturation. And this is exactly in your wheelhouse of everything oligodendrocytes, right? It's got this node of SOC6 as serving as like a molecular brake. It's a really cool concept. I was raving about it when we covered it a couple weeks ago. It had a little bit of everything in this paper, from basic discovery to translational applications, just like what you were saying with the emphasis of your lab. Could you actually walk us through the paper, talk a little bit about what you meant with gene melding and what you found when you actually experimentally released that SOC6 break? So talk about this paper.
Yeah, well, appreciate you covering it on the recent episode, and excited to dive in a little bit deeper here. And as you noted, this paper has a lot of different approaches in it, and as you can imagine, was really a confluence of a lot of different efforts in the lab over a number of years. I think the origins of this paper probably predated sort of 10 years ago or so, where we really started to think about how this transcription factor SOC6 may be playing a role in oligodendrocyte development, and ultimately led us to understand that it has this very special role in controlling the timing of maturation. And it was led by Kevin Allen and Jesse Zahn, as well as Tyler Miller when he was in the laboratory, really thinking about what are the key regulators of not only that early step in differentiation, which is something as stem cell scientists that we all think about, or what is that key trigger that is driving that cell specification event from a progenitor cell, in this case, the oligodendrocyte progenitor cell, into a differentiated phenotype. And we think a lot about, as a stem cell field, we all see the Waddington's epigenetic landscape map with the marbles, rolling down a hill, and we think, okay, you make that cell fate decision, and you roll down the hill. But what we actually found in this study is that there is a governor or a regulator of the timing of maturation, and we think that this is fundamentally a really exciting discovery that may extend into other cell lineages as well. But in this case, identifying sort of a timing mechanism that regulates not that early specification event, but actually when the cell will mature. And so if you think about it in the context of this Waddington's epigenetic landscape map of the marble rolling down the hill, it's sort of like there's a sort of road at the middle where you can be in a holding pattern, right? And the cell has the ability to govern that timing so that it further develops or further matures at the right place and at the right time. And so we found this out by a number of studies, but I think most importantly, when we were looking at the epigenetic chromatin maps across development, we saw this really unique and interesting pattern in immature oligodendrocytes at a very defined set of loci. And when you look at the histone acetylation maps, K27 acetyl here, or ATAC-seq, you had this like very strange pattern that looked like you were getting signal across the entire gene body. And at first you think, well, maybe that's artifact. And in fact, reviewers, pressed us a lot to show this definitively, and we're very grateful for the feedback and insight that they provided to make this manuscript better. But ultimately, when you think of a transcription factor, you don't think of it binding across. You don't think of seeing signal across an entire gene body. And so that was very peculiar. We ultimately identified SOC6 as being a key player at this 100 or so sites in immature oligodendrocyte. And what we think is happening there is that this term, this sort of sexy term in the title of gene melting, is that this is really a sort of a biophysical transcriptional unit where you have extreme chromatin decondensation, right? You're losing the contacts everywhere, and you have just very high transcriptional output. And it's not a liquid phase separated condensate like we see in other contexts, but it is this biophysical transcriptional unit that SOC6 is a part of. And then when you remove SOC6, and likely if you were to remove other components of this transcriptional unit that we have yet to define, right, that whole thing collapses, and then the cells mature. Right? But it's this very exciting and unique idea that you are positively regulating this break on maturation until the cells are ready and in an environment that is appropriate for maturation.
Such a brilliant and innovative conceptual advance, and I'm glad you filled in there, because it shows how like an idea that's the genesis of which is a decade ago, and over the course of many postdocs and techs and at all levels, collaborators working on this thing, you kind of peel the onion, right? And your rendering there really struck home not just how you got there, but what the impact, and as you said at the jump, like how this conceptual events may apply in other cell types. So yeah, of course, we love that story. We were big fans when we covered it. Because as you described, it had all these many dimensions, literally this temporal dimension of maturation, but all that biophysical, as you described, and the practical insight into like the oligomaturation being the rationale, but also that concept, right, in terms of the epigenetics and that active breaking, and all within this framework of a therapeutic approach, right? Kind of bearing the lead, a therapeutic approach to promote myelination. And the discovery of that element, as you described also, was powered in part by these genome-wide microRNA screens. And that's an approach, these screening approaches, you've leveraged over and over across your model systems. And so staying on that, but shifting gears a bit, I want to focus on this other story in Nature Neuroscience that kind of took a different angle on disease, utilized a screening approach to identify these relatively ubiquitous environmental chemicals that impair oligo development. Tell us a bit about that as a kind of counterpoint to this therapeutic, how do we repair, but more like documenting this battleground within which we live, where there's all these, hostile environmental influences, and what the meaning of that is, both for neurodevelopment as well as disease progression. Like, what's big picture? What are you getting at there?
Dalen, I appreciate the question. And this, you're referring to a paper led by Aaron Cohen in the lab, who is an MD PhD student that we published in Nature Neuroscience last year. And in this case, we really leveraged our high throughput screening, cellular screening approaches to understand particular chemicals in our environment that might be able to either impair oligodendrocyte development or regeneration, or more directly to which oligodendrocytes are exquisitely sensitive. And so the rationale here is to understand, are there environmental drivers that might be either initiating in the context of, early developmental disorders, or exacerbating, adult neurological or neurodegenerative diseases like MS. So you could imagine that there's some underlying initiation or predisposition to particular disease, say MS, but in the context of exposure to a certain environmental driver, say a particular household chemical, that could be further exacerbated. And so to get at whether or not there might be chemicals in our environment that are driving oligodendrocyte dysfunction or loss, we collaborated with the EPA and used their library of environmental chemicals of concern, their so-called ToxCast library, and we were able to screen nearly 2,000 compounds or chemicals that are found pervasively in our environment, most of them found in all of our households, and understand their impact on oligodendrocyte development. And we identified two classes of compounds, but one in particular, quaternary ammonium compounds, which are found in common disinfecting agents to which oligodendrocytes were uniquely and exquisitely sensitive. And quaternary ammonium compounds were identified as being cytotoxic to oligodendrocytes in vitro and in vivo in mice, as well as in human brain organoids as well. So this really set the stage for thinking about how these chemicals could initiate or exacerbate neurological diseases where loss of oligodendrocytes might be involved.
Yeah, I mean, it's right in the wheelhouse of exactly what we're talking about in terms of the expertise in your lab. And kind of expanding on that, I wanted to get your take just more broadly on technologies utilized for high throughput screening. I mean, you mentioned some of the work that you guys do, like especially when it comes to deep molecular profiling, different chromatin states and all that. How do you see the trade-offs between scale versus depth in the different screens that you design? I mean, this is something that I run into a lot with, like I work a lot with iPS cardiomyocytes. So, molecular screens and drug screens and iPS cardiomyocytes, you can get super broad and have massive compound libraries, but then you may not go deeply as much into each of the mechanisms of how those compounds are actually influencing, say, cardiomyocyte function. So talk about kind of the design of these screens and what do you look for there?
Yeah, I mean, I think on its face, we like to screen we like to screen everything, and there's many jokes in the lab about if we can get a library to something, we'll put it on our cellular assays. I think more sort of specifically here, we like to develop cellular assays, again, mostly focused on oligodendrocytes, but we have expanded that purview out into other glial cell types, including astrocytes and enteric cells. The thing is, we like to mimic one specific key aspect of either development or disease. And so we have a number of cellular assays in the laboratory focused on glia, oligodendrocytes predominantly. And we like to screen relevant libraries that might be able to provide, key insight into whether or not particular targets within those cells might be amenable to changing that particular cellular readout. So in the case of, can a compound stimulate oligodendrocyte differentiation? Like that's something that we've screened basically every library that exists for. And to answer your question about sort of scale versus depth, we try to strike a balance, which is to use initially, whether it's genetic screens, but in particular on the small molecule side, libraries that are big enough to have breadth across the druggable landscape, but where most of the compounds in there have canonically annotated targets, right? So you're not just doing this to screen 1 million compounds and say which is the best one that we might be able to to put into a patient. that's really challenging because it's hard to delineate mechanism out of those screens. But if you can use a bioactive library where all of the targets at least have a canonical annotation that provides you with an immediate understanding of particular pathways or particular nodes that might be the key drivers of your phenotype. So I like to use these screens as a hypothesis generator. In most cases, even though you hear drug screen, you think about, okay, you're screening for a therapy. We are typically not screening, at least initially, for a therapy. We are screening to identify, a set of targets that, when manipulated, will change your phenotype either positively or negatively, and that mechanistic insight allows us, really to define what's going on. And then if we wish to come back on top of that with a therapeutic development program, we've identified a target and we can tackle that target with maybe the same modality, but possibly a different therapeutic modality that might be better served for manipulating that target in preclinical models.
Yeah, and it's super robust, right? In terms of like if you had to how you design a lab, it's a great template because really at the bottom of it all, you're trying to answer questions, and while the rationale for the studies may be geared towards therapeutic application, ultimately you're tinkering, right? And you're unpacking mechanisms that have intrinsic value just in the knowledge. But then, as you said, you circle back, right? And you said that in your intro of like what the lab focus is. And, of course, you have all these stories that are deeply mechanistic and novel, seminal from the early days, right? And epiblast stem cells, like you were kind of getting at, fundamental cell states from a mechanistic standpoint. But as you said in the intro, you've been very deliberate about trying to bring those insights into treatments, right, for patients. And, not surprisingly, it's almost 10 years ago, you started Convello Therapeutics, which is a biotech focused on, and I'm quoting, unleashing the central nervous system's natural healing potential. And this is a really exciting phase of this translational push that we're in the midst of cell therapies, going into clinical trial across the board. Some of the most prominent trials are in the neural space. So tell us that, put on that hat for a second for our listeners. Tell us about the experience of founding and guiding a biotech, and also like specifically, what's your vision for Convello moving forward?
Yeah, I think this is a great conversation to have following our prior topic here. And so the foundation of Convello, that our initial publication in Nature in 2015 was a phenotypic screen on a small a library of FDA-approved compounds for their ability to stimulate the generation of new oligodendrocytes from the progenitor cells. So we developed a scalable technology to grow oligodendrocyte progenitor cells in the lab in 384-well format, and to do phenotypic screens for compounds that might stimulate the generation of new oligodendrocytes. And the hypothesis here was that in diseases like MS, and now we know in other diseases, oligodendrocytes are lost, but in particular in MS, where the immune system attacks and destroys oligodendrocytes, there is a need to regenerate or replace oligodendrocytes. I think what's cool about the brain and oligodendrocytes in particular is that you have an endogenous pool of progenitor cells in the brain called oligodendrocyte progenitor cells, that their job and their role is to be able to regenerate new oligodendrocytes. And so it's pretty unique in the brain. There's not a pool of progenitor cells that serve as a regenerative cell type outside of OPC. So these are a really unique population. And we thought that, one approach could be to transplant in new oligodendrocyte progenitor cells. And I think that has a lot of potential. And hopefully we'll move into the clinic at some point in the near future. we took the approach that there was an endogenous pool of cells, and that by unlocking or unleashing that endogenous potential, that we could identify potential nodes of intervention that we could stimulate the enhanced regeneration of new oligodendrocytes. So we published a paper. We identified this particular structure, which was an imidazole structure, in that paper that had really potent ability to stimulate the generation of new oligodendrocytes. Now, that imidazole structure is commonly found in topical antifungals. And so it's really a weird finding, right? But when you do a phenotypic screen, you don't always really understand the direct mechanism of action, but we wanted to show in vivo that those molecules could stimulate remyelination and reverse paralysis in mouse models of MS. The headline of BBC News for that article was like something weird, like, athlete's foot drug may cure MS, and there was a picture of a fungally infected toenail, which was like the media release that went along with it. And so you of course are very proud to have anything on BBC News, but it was pretty funny. But ultimately, I think, as I'll come full circle here in a second, the derivatives of that work may ultimately prove to be true here. So we went on with our colleague Drew Adams. So Drew Adams was recruited around that time here to Cleveland, to Case Western Reserve University School of Medicine, from Stuart Shriver's lab at the Broad. Drew was a Harvard trained and David Evans lab, or synthetic organic chemist. And so he brought this sort of combination of hardcore chemistry and chemical biology with my lab's glial biology. And ultimately in 2018, we published a paper with Drew's lab identifying the therapeutic targets for these and other small molecules that are driving oligodendrocyte regeneration and remyelination. During that time period, we founded a company, Cambello Therapeutics, that we started here in Cleveland. Cambello's raised $32 million in equity financing and had significant additional funding from a strategic partnership with Genentech and Roche. And excited to say now that the derivatives of those early stage studies have now completed IND enabling studies, and our Cambello is set to file an IND later this year and expecting to start clinical trials next year. So, it's been a really exciting story, going from an early stage phenotypic screen on a small set of compounds to delineating the mechanism for how those compounds are driving enhanced oligodendrocyte regeneration and remyelination in vivo, and now with the opportunity to see the science that was done at the benches out the door here but behind me have its chance to potentially benefit patients. So it's a pretty exciting time for us, and really excited about the future for this program.
That's awesome, man. Congratulations.
Yeah. Living the dream. I mean, I think that's the dream for a lot of stem cell biologists, basic scientists these days, is to take that phenotypic discovery, that initial observation, and bring it all the way potentially to the clinic through a drug discovery pipeline. I mean, that's super cool. It's something that I'm striving to get towards as well. So, in addition to your role as a PI, biotech founder, you name it, a little bit of everything, you've also, been a great mentor. You've mentored a large number of trainees who are now building their own careers in neuroscience, stem cell biology, regenerative medicine. And like I mentioned before the show, a while back, we chatted with Dr. Marissa Scavuzzo, who did her postdoc in your lab and also has her own lab now. So just, trying to pick your brain a little bit as a fellow PI and as a new PI, I'm always really interested on the strategies behind mentorship, right? So what principles guide how you kind of run your lab or support your trainees, guide your mentorship principles? And I guess since we're sort of in a new reality right now when it comes to the realities of funding and just everything that's happening right now, I feel like mentorship has to evolve in some ways, because not everybody's going to follow in our footsteps and become an academic PI, right? So how do you balance all of that? So tell us more about your style of mentorship.
Yeah, appreciate the question. my excitement and enthusiasm for mentoring the next generation students and trainees at every level, from postdoc all the way down to summer high school and middle school students who are just here to shadow for a few days, is why I'm sitting in this chair. Obviously, I'm very fortunate and lucky to get to play in multiple sandboxes with the entrepreneurial activities that we've had, some of which we just discussed. But the opportunity to participate and be part of helping the next generation move forward is why I'm sitting in this chair and not permanently in a chair at a biotech or pharma company, because I absolutely love that aspect of my job. And I think, to your point about changing style, I would just say that I don't know that we need to adapt as a field, but I think we need to adapt individually to each student. And so that's really I think a key component of my mentoring style, which is to help and advise each individual person, understanding where they want to be. Not everyone wants to be a PI in an academic environment. And in fact, I would say many of the folks, coming to my lab are very excited about going into pharma and biotech. And you have to be very intentional and purposeful to meet their goals and to help them, get to a point where they can move on to that next stage and not fit them into the academic, track bubble, which I think is something that often we try to do, and maybe our system sort of forces us to do at times. But I think if we can get outside of that and provide training environments that are more broadly open to multiple career paths, I think we'd be better served in our laboratory and continue to have a growing population of students and trainees to populate all of our laboratories. And I think the only thing, I don't think that I do anything special other than to care and to understand where each individual wants to be. But the one thing that I really sort of force is that when folks come into my lab at any level, that they have to define and work on their own project. And so while it may or may not be apparent to the trajectory of the lab over time, is not me saying, okay, now you work on this next step, and then the next person, you work on this next step. It's a little bit of a narrative wrapper around all the cool science that's been done by the amazing folks in the laboratory. But when folks come in, graduate students, postdocs, it's, what do you want to work on? What are you excited about? And what are the technologies and expertise that we have in our group, in the larger Institute for Glial Sciences here, or within the medical school, that you can take advantage of to answer a question that's exciting to you? And sometimes people do not like that. And those people don't end up staying past their rotation in the lab, and that's okay, right? Folks want a prescriptive project that they can follow the next step, they can publish paper and move on. Jumping into the deep end and sort of literally floundering for a year, trying to find your space, trying to find a cool idea that no one else has worked on, something that you think, in 4 years when you go to publish it, will still be innovative and novel, is challenging. But that's what you do for the rest of your career, is to sort of figure out what data you have, what's going to be interesting, and try to put a narrative and a story around it. And so I think that's been the most exciting, is all the projects in the lab that have come in. For the most part, I've personally, I've not known the underlying either technology or specific field that was brought in by the trainees, and we apply our favorite cell types of interest and the techniques and expertise that we have in the lab to really answer a unique question. And so I get to learn along the way. And it's just fun to come in every single day and be part of this amazing team.
Yes, man, living the dream for sure, as Arun said. But your mentees, your trainees are lucky to have the benefit of your broad experience in wearing all those hats for sure. And as a lot of, I think, PIs describe, you need that young talent and that new technical expertise, right, to reinvigorate your tool set. Um, but you do all you do is care. I mean, you make it sound like it's not a lot, but it is. And that's the best, I think. Not mentors or mentees, but the best labs, I think, are like a family, and like any leader of the family. In this case of a lab, you lead from within, I think, is another great part of your mentorship. But you care about the outcome, right? Because these are your so-called children, just like you would your own kids. You want them to be the best scientists that they can be. And so you have certainly done that, and you stayed close to home in doing it. You're an Ohioan down to the roots, born in Cleveland, and apart from a stint in the NIH Oxford Cambridge Scholar Program, you spent most of your research and teaching career at Case Western Reserve University School of Medicine, which is an institute with almost 2 centuries of history. You can't sleep on Case Western Med. But tell us what you love about the Institute. What's kept you there so long, brought you back? And what do you love about it and the way science and medicine are practiced there?
Yeah, I mean, as you said, Cleveland's home. Grew up here. Did my undergraduate work at Case Western Reserve University. Met my wife Anne while we were pursuing our undergraduate studies, and then was off in Oxford and at the NIH for graduate work. And I moved literally 3 miles from where I grew up. So pretty close to mom, and which has been a great opportunity for her to grow up, for my daughter to grow up, near her, and it's just been incredible. So my roots are here. Cleveland's always been kind to me. The university's always been super supportive of me. I mean, I think what I really love about Case Western Reserve is the people, and it's this sort of Midwest mindset. It's a very collaborative, interactive, we like to say blue collar, working class town where people care about doing the right thing. They care about each other. We care about families, and so we work hard and we play hard, but it's just really a tight knit group. I think what also drew me back here outside of that is we're immersed in a world-class healthcare system. Cleveland Clinic, University Hospitals, our VA is the third largest in the country. And so, being at that collaborative center of medical excellence, it really inspires someone who's a PhD scientist, but who has really a strong translational drive to be able to take our science and bring it forward into patients. And I think I've really enjoyed the students. I've run my lab predominantly with graduate students, and that was purposeful at the beginning. But as and as we've expanded out to having a number of postdocs in the lab, but our graduate program here is outstanding. Our MD PhD program is the oldest in the country, and I've really enjoyed the opportunity to have translationally focused, interested students really drive these innovative projects that have formed the core basis of our laboratory over the last 15 years.
Yeah, don't sleep on Cleveland, don't sleep on Ohio, that's for sure. And as somebody from the middle of the country, flyover territory, I suppose, I can definitely appreciate that. And I had the opportunity to visit Cleveland and Cleveland Clinic in particular a few years ago, and I was really blown away. I mean, as a cardiovascular biologist, Cleveland Clinic is phenomenal when it comes to everything heart related, as you know. So it's a great place to be, and it's It's cool that you're able to kind of come back to your roots in that way. It's not always the case. I mean, I grew up in Alabama, so maybe one day I'll I'll head back that way. Thanks so much for sharing your journey, Paul. It's good to catch up with you. And before we let you go, we've got a couple of peripheral questions that we'd like to ask our guests here on the show, just so our listeners can get to know you guys a little bit better. So first thing, if you were not a scientist, what would you be?
Well, I mean, growing up, I had two clear career trajectories. One was to be quarterback for the Cleveland Browns, and the other was a veterinarian. So I'm gonna just take a flyer here and say that Joe Flacco and Dylan Gabriel and Shedeur Sanders aren't gonna welcome me into that quarterback room right now. So I think I'm gonna go with veterinarian would be my answer.
You might do a lot better than some of those quarterbacks.
Yeah, I was about to say, might be an upgrade. I don't know.
We're having a rough go, but I think they're going to figure it out eventually. At least that's the perpetual Cleveland sports fan hope.
Hey, fingers crossed, fingers crossed. Last question for you. What's the biggest misconception about science that you want to resolve?
I think a common misconception and something maybe that's being a little sort of weaponized right now is that like science delivers truth with a capital T, right? I mean it's like science is this perpetual building accumulation of facts. I mean that give us conclusion based on what we know at the time. And new data shifts that view, right? And sometimes significantly reshapes our conclusion, and that's okay, right? That's the whole point of the scientific process. It doesn't mean that the prior work was wrong or fake. It's this continual pursuit of knowledge, and I think the more we can engage and have an understanding of what science is and what the scientific process, I think the better broad appreciation for what scientists do in society will be had.
That's such a great point and well said, right? Not a collection of facts, but an apparatus. And it's sad that by trying to make it and weaponize it in part as a set of facts that you've kind of dismantled the apparatus. And you talk about your students, and we talk about the trainees all the time on the show because that's it. That's the engine. And the idea that some of these young minds that have the imagination and creativity won't ever get the opportunity to see the thing for the first time, right? To get that bug and plant that seed, that's the tragedy. So I'm glad you mentioned that, and I'd like to underscore it. It's a method. It's just a way of doing things that's predicated on the idea that we don't know, and the joy in figuring it. So a lot of things you said, a lot of kernels today, Paul. It's such a great joy talking to you, and thank you so much for sharing. It's always great running into you at the meetings because you're just a level, steady influence. I'm very mercurial all over the place, but you bring me right to center, and I appreciate your influence in my life. And I hope that our listeners are going to share in that same joy and appreciation. Paul, thanks again.
Daylon, Arun, thanks so much for having me today. I really appreciate all that you do for the field with this podcast.
All right, you guys, that brings us to the end of this episode, a stirring conversation with Dr. Paul Tesar. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCellPodcast.com, or via email at info@STEMCellPodcast.com with feedback or to suggest guests. Until the next time, which should be coming at you in a couple weeks, thank you so much for listening.